Another thing you could do, honestly, like I said before, if you just like don't have ideas of what you could write about, maybe you can meet me online and we can just talk. And I can talk to you about your interests and things like that. And maybe since I'm the biologist, and if I can get to know you a little bit better, I'm like, hey, maybe you consider writing a paper about this or that or the other thing. You know, let me help you. And then before you submit it, again, maybe meet me online. Let me look it over and I can talk to you about it. Like, oh, I'm looking at this and your source does not match what you said here. Or, you know, all these different things I can help you before you even hit the submit button. Anyway, Aki. Not here. Boggs is here. In WVSU here. Bonner is online. I'll remind you, if you're online, you have to send the words in. Just because I see that you're online doesn't mean you're present. Anybody can log on and then go back to sleep. So please send the words. Clay is here. It's a little dark. Is that Clay? Clay? Okay. Sorry, it's dark and I'm not wearing my glasses. Clay is here. Dawson. Here. Yep. Thought I saw you back there. Ellis. Ellis is not here. Paula is not here. Green. Green is not here. Harrison is here. Henson is not here. James is here. If you're at WVSU here, you have to tell me because it's so dark today. I can't see. But anyway, James is here. Kesterson's here. WVSU here. King is not here. Lewis. Thought I saw Lewis. Nope, no Lewis. Linda Q is here. Up in WVSU here. All right. WVSU Wednesday. Morrison is here. Grace, is that Grace? Yep, Grace is here. Ward is back there, right? Yep. Ward is here. Wilcox is here. In WVSU here. Go State. Wilder's online. Once again, if you're online, you need to send the words to get your points and to be counted as present. I thought I saw Parks come in earlier. Yep, Parks is here. Someone else came in while I was doing that. Was that Paula? Paula here? Yep. Did I not get anybody? Did I not get? Aki? Yep, you came in. Sorry. So Aki is here. In WVSU here. And then I said Paula is here. And then we had somebody come in. Is that Ellis? Yeah? Okay. And King is also here. Of course, we're 2 minutes late. That's okay. Not you. I'm 2 minutes late. I ran behind a little bit. That worked out for those of you who came in late. So let's get this party started again. Close the door. Reminder, we have lab today. Everybody is coming. Well, everybody is eligible to come, which is exciting. I think we've all figured out the whole rigmarole with labs and pre-labs. Oh yeah, speaking of which, somebody asked this question at the end of class yesterday, so you didn't all hear it. There is no pre-lab due on Friday because next week's lab is just going to be a pre-lab lecture. It's such an important lab that we're going to have a pre-lab lecture next week. And then the week after that, we're actually going to do the lab. So technically your pre-lab is not due Friday. I'll send out an announcement to remind everybody with a link for the pre-lab because if you want to, since you still have a full week to do it, it's not due until next Friday. I recommend getting started now. But anyway, we do have lab today. Make sure you wear closed-toed shoes and no shorts, right? So you do what I do. Bring some pants with you if you need to change into them. Anyway, any questions about lab? Make sure you're on time. The doors close at 10. Mr. Ward?
Next week's lab, are we showing up here at
the
lab time to do the lecture?
Exactly. For you guys, it's essentially the same. You're still going to show up here at 10, and then, yeah, good question. And I'll make sure I put that in the announcement. Any other questions? All right, so we left off talking about the different types of potential energy. The first word for attendance will be lake. I'm going to presume that's a lake they're swimming in. I don't know if it is or not, but anyway. First word for attendance is lake. As I said, if you download this PowerPoint, you can kind of watch this little animation that has some energy concepts. Here's an analogy I like to use. So you're going to see this picture pop up a lot. at least at the crossbow. And I'll explain the pictures here in a second. Let's talk about the words first. Moleculars have a form of potential energy called chemical energy. And I told you that earlier when I first introduced you to the word potential versus kinetic energy, right? I said kinetic energy is the energy of motion, right? Energy of movement. I said potential energy is stored energy. And I said you probably already do that. You've been taught that probably since kindergarten, right? That's old concepts to you. Then I got a little bit more specific. And when I said that stored energy is either due to location, which like I've said the other day, this marker has more potential than this marker because they're identical, other than the fact that this one's higher, right? So this one has more stored energy. So we're all very familiar with the concept of stored energy due to location. But then I introduced you to a different concept, was the concept of stored energy due to structure, right? And that's what we're getting at here. Chemical energy, that is potential energy due to the structure. So once again, however much energy it takes to build a molecule is how much, generally speaking, as far as we're concerned, however much energy it takes to build a molecule is how much energy is released when you break that molecule. So in a sense, when you think about it that way, the harder it is to build a molecule, the more energy is going to be released when you break it. Also, as a side concept, and this part's a little bit less important, but just so you know, actually, you know what? I won't even say that. So yes, think about that. Try to remember that. However much energy it takes to build a molecule, as far as we're concerned, is how much will be released when you break it. So this is due to the arrangement of the atoms in the molecules, and it can be released by chemical reactions. Now, here's what I'm trying to get out with this picture. How many of you ever try to put two magnets together when they're both, the same polarity, right? What do? They repel each other, right? They push each other away. The same sides of the magnets don't want to be next to each other. Well, let me ask you this. What's on the outside of an atom? All atoms. What's on the outside of an atom?
Electrons.
Electrons, yes. And what do they charge? What are their charges?
Negative.
Good. Negative. So the outside of all atoms have negative things on the outside, right? They have a negative force field, if you want to call it like that. That's not the actual term, but you could think of it like little negative force fields. So when you think about it, when these two things come together, they're kind of repelling each other, right? Because that's a negative thing and a negative thing. In a sense, based strictly off of that, these atoms don't want to be together because those electrons are repelling each other. So even though we've learned about covalent bonding and ionic bonding, where they're sharing electrons or giving them up and taking them, even though they do work together with those electrons, at first, at least I'm very oversimplifying this, but at first you can try to envision them repelling each other. It's going to take some sort of energy, even though they eventually do want to be together, because that will eventually put them in a lower energy state, which is something I'll talk about later. It's going to take some energy to squish them together. Right? So even though they want to be together, in the end, at first it's going to take some effort to get past that negative force field, if you will, right? So that's where that energy is coming from. How much energy it took to squish those things together is how much energy will be released when you break that molecule. And then that's the reason I'm using this as an example is, however much energy it takes to pull that crossbow back is how much energy is going to be released when you pull the trigger. Does that make sense? And generally speaking, as you might imagine, the harder it is to pull that crossbow back means that's more tension, right? There's more energy there. So the more energy you have to put into that thing to cock it means it's going to be more powerful. More energy is going to be released when you pull the trigger, right? Because there's different types of crossbows. I'm not expecting anybody to be an expert on that, and I'm not either, but some of them are easier to pull back, some of them are harder. So that's a concept we'll keep coming back to, that crossbow concept. Any questions so far? All right, so again, this is not written in your textbook, so I'm not specifically going to ask you all these things I'm telling you, but if you understand these concepts, then you can really understand the things I'm going to teach you later. They are from your old textbook, or the old textbook. So a good analogy that's not in your textbook but is in the old textbook is that cells and engines use the same basic process to make chemical energy that are stored in fuels available for work, right? So for us, we eat food, right? And there's chemical energy stored in the molecules of our food. We break those molecules, we release that energy, and that's how we live. And engines aren't that different, right? So engines, you put in some octane. That's powerful, not powerful. It's an energy-rich molecule. When you break it, it releases energy, and in that case, the energy is used to drive your vehicle, right? So I like the analogy that the old textbook did there. And here's a picture showing what they're talking about. Another interesting thing, not that I'm going to ask you this, but it's interesting to me that, all right, so to move the engine, right, you need to burn octane in the presence of oxygen, or break down octane in the presence of oxygen. And then the byproduct of that is carbon dioxide and water. Not that you need to know that. You will need to know this eventually, but we'll come back to it. But it's funny to me, or interesting, because it's the same thing that happens in your cells. You break down glucose instead of octane. You break down glucose in the presence of oxygen. That releases energy, and your byproducts are carbon dioxide and water. So similar thing when you look at it that way. That brings us to the definition of cellular respiration, and I'm not going to ask you the definition of cellular respiration. There's going to be a lot of questions on this exam when I say, well, first of all, let me tell you this. I'm skipping ahead just for a second. Cellular respiration is broken down into 3 stages. Not that you don't need to write that down yet, because we're going to talk about it. But most of the questions on the exam that are about cellular respiration are going to be, in which stage does this happen? In which stage does that happen, right? So I'm not going to ask you the definition. But most of the questions are I'm going to say, this thing happens, and then I'm going to ask you which of the 3 stages that thing happens. But anyway, what is cellular respiration? That is the energy-releasing chemical breakdown of fuel molecules. And again, I'm not going to ask you the definition. Again, some of you really need some independent work topics, so here's something, and one of your classmates has been doing this, and I think this is one of the good things you can do for independent work. Fact check me, all right? Don't take my word for it. So even though I'm telling you this is the definition of cellular respiration, you could look it up and say, well, okay, according to this source, they define it as this, and I'm sure it'll be the same definition, just worded a little bit different, right? So again, you can just email me that. Don't do it now, pay attention to the lecture. But afterwards, you can just send me an email and say, I looked it up, and according to this source, this is the definition of cellular respiration. And then boom, as long as it's correct, you get a point, right? You don't even have to write a paper, just answer a question. And in that case, since you're just answering a question, you don't have to worry about plagiarism. You can use the words directly as written by your source. Anyway, that's what cellular respiration is. The storage of that energy, so again, we're breaking down the fuel, we're releasing energy, and then our cells take that energy that was released and they're storing it in a different form. And we'll talk about what that different form is later. Here's a number you do need to know. I'm not big into numbers, but this one you need to know. This one will be on the exam. About 34% of food energy is converted to useful work. About 34% of food energy is converted to useful work. The rest of it is released as body heat. You might say wasted, but not necessarily, right? Because we need that body heat. So the reason you are 98.6 degrees is this right here. It's because you're breaking down fuel, and only 34% of it's being put to work. The rest of it is used to just make you warmer. If anybody doesn't know what that picture is, it's like an infrared picture. So the hotter somebody is, you can see the different hot spots on their body. Anyway, any questions about this slide? And again, another good independent work topic. This number is according to the old textbook. So once again, you can just look up and answer that question. Question being, how much food energy is converted to useful work in your body? See what another source says. Send me an email. According to this source, it's actually exactly 37%, or something like that. Who knows? Or maybe they'll have a range. It's 32 to 35%. It could differ. I'd be curious. Anyway, any questions about this slide? All right, so again, as far as the exam is concerned, some of that bottom bullet point is important, because this other stuff is kind of an introduction to what we're going to talk about later. All right, here's some stuff that's from your textbook. I'm going over it because it's in your textbook. But these are, and I might have a couple questions, but it's only going to be, if I have any, it'll be like 1 or 2. So if this is, that shouldn't be too confusing. But if it is too confusing to you, you don't need to worry about it. Let me say this. I mentioned earlier when I told you what entropy was on Monday, I said as far as we're concerned, entropy is very unimportant, right? But I said if we were doing this for real, and we were doing chemical reactions where we keep track of energy and entropy, and we do it mathematically, that's when that concept is important. Same here. Free energy is very important if we were doing this mathematically, but we in this 100 level class are not. We're also not worried about delta G, which is the change in free energy. So I'm going to talk about this because it's in your textbook, but just know I'm not really going to ask you any questions about it. So before I even say anything, first of all, let's talk about what free energy is. Free energy, which is signified as a big G if we were doing this mathematically and have an equation, but we're not. That big G quantifies the energy available for doing work after accounting for losses. Which is another way of saying when this chemical reaction happens, when this plus this gives us this, free energy is saying this is how much energy is now available to do work, right? Whether that be heat energy or different types of energy that we'll talk about later, that's how much energy is released, so to speak, right? So with that concept in mind, and again, mathematically that's important, but for us it's not that important. What we're saying here is that there's two different types of reactions, exergonic and endergonic. So exergonic is where delta G is negative, and I know that sounds really weird, and luckily for you guys, we're not doing this mathematically, so that part doesn't matter to you, but here's what you need to know, if anything, for the exam. Exergonic reactions is when energy is released. And it's kind of in the name, X, right? Like it's going out, it's exiting. Energy is released in exergonic reactions. Another way of saying that, and again, I'm only saying this because it's in the book, so you can brain dump it after I say it. This means that the products have less free energy than the reactants. Meaning, if I say A plus B gives us, gives us the molecule AB, right? And what that's saying is, in this case, if it's an exergonic reaction, the products right here have less free energy than the reactants, right? So I don't want to put it that way, because the energy has been released to do work, right? So there's less energy available in the products. But again, the part you need to know for the exam, if anything, is just this. Exergonic reactions release energy. And as you might imagine, the opposite is true of endergonic reactions, which also hopefully makes sense to you because it almost sounds like it's in. The energy is coming in. That's when energy is absorbed. So again, for the exam, if anything, you just need to know that exergonic reactions release energy. Endergonic reactions use energy. The other stuff is not important for a 100 level class. And I'm only putting it up there because your textbook talks about it, so I wanted to cover it too. That way, when you're studying your textbook and you read it, you don't think, what in the world? He didn't say anything about this. Another thing worth mentioning, even though this won't be on the exam, is that these exergonic reactions generally happen quickly, not quickly, but they're spontaneous, meaning, again, they're releasing energy, so they don't necessarily need a large input of energy. Obviously, endergonic reactions do, because by definition, they need energy, right? So these are non-spontaneous. They actually require an input energy for them to start. And here's the term you do need to know. This last bullet point will be important. We will use this term later. There will be questions on the exam, not where I ask you what activation energy is, but when I say they'll involve activation energy, so you need to know what it is to answer the question. All reactions require an initial energy input known as activation energy. So activation energy is the initial energy input. Oh, this makes me a point. This is a question a lot of people list on the pre-lab this week. What is diffusion? So think about what you write, because a lot of people wrote, like there was a sentence in the pre-lab that said, molecules move back and forth in a process called diffusion. And then after that, it goes on to define what diffusion is, but people just wrote the first part, which is that molecules move back and forth. Like, yeah, but that's not diffusion, right? They also move back and forth in other ways. But then if you look at the comma, definition, right? So same here, right? So if I were to ask you what activation energy is, the answer wouldn't be all reactions require initial energy. No, let's get on to the right spot. The activation energy is the initial energy input, right? That's what it is. The initial energy input. So here's an example. And this is another definition of activation energy. Same way of saying, same thing, saying in a different way. Activation energy is the energy invested to start a reaction. And again, I'm using this example. There's a lot of energy in that crossbow because you cocked it, right? You pulled it back, you put energy into it. However much energy it took to pull that string back is now ready to be released. But that thing's not going to be released. It's not going to shoot itself, right? Even though that is an exergonic reaction waiting to happen because it's going to release a lot of energy, it still needs an input of energy before you can happen. In this case, that's pulling the trigger, right? Think of also, I'm really bad at drawing, especially when I'm trying to use a mouse. Think of a hillside with a big rock on top of it. Huge boulder. That thing's got a lot of potential energy, right? What's going to happen if it rolls down, if it starts moving forward? It's going to roll down the hill, right? Get faster and faster and release a lot of energy. But it's just sitting there. It's not just going to roll by itself. It needs some sort of energy input to start that very exergonic reaction. Yeah, it's going to put off energy, but it still needs some sort of input. Maybe a person to push it, or an earthquake to shake the ground, or a really strong wind, whatever it may be. These reactions, even the ones that release energy, still need an input of energy, and that is called activation energy. One more picture that shows this, and then maybe I'll take questions about it. This is from the, two more pictures, I'm sorry. This is from the old textbook. So again, reactants, right? This is an energy, this is a reaction, both on the left side and the right side, both are the same chemical reaction. Both of them are releasing energy because the products are lower energy than the reactants. We've released energy. But this reaction requires a lot more activation energy. And again, trying to picture that like a wall, and literally you're just trying to push a dumbbell over a wall, right? That's going to take more energy for you to get it up over that wall before you can finally drop it. Now, once you drop it, that's the easy part. But here, the activation energy has been reduced, so now you only have to pick it up a little bit. Does that make sense? Like literally just try to imagine this as a building and a set of dumbbells and you trying to throw them over a wall, right? For some reason, you're on top of Wallace Hall and you've got a set of dumbbells, and there's a wall this tall. I'm like, oh, okay, that's nice and easy, right? Or there's a bigger, taller wall, and you got to do it like that. Either way, you're going to release a lot of energy when you drop a set of dumbbells, right? But when you have a smaller wall to get it over, you have less energy that you needed to put into it. That is called activation energy, right? That wall represents activation energy. And now, finally, the most important thing for this concept that I'm teaching you is that this is what enzymes do. This is the purpose of an enzyme, and that will be a test question. What is the purpose of an enzyme? Is to reduce the activation energy. The purpose of an enzyme is to reduce the activation energy. So chemical reactions, again, now we're looking at dumbbells and stuff, but again, now let's try to focus back on chemistry. Chemical reactions, even when they release energy like this, they require an input of energy. Enzymes, for different reasons, depending on the enzyme and the specific chemical reaction, lower that amount of energy that you need. And again, let's go back to my original analogy earlier today. I was saying, when you push two atoms together, they don't want to be together, right? Because they have negative things. They have those negative electrons on the outside that repel each other. So it's going to take some energy, even though they do eventually want to be together. First, you got to get through that negative force field, right? So you need some energy to push them together. That's the activation energy, that energy to get them past that spot, whatever it is that's keeping them together. And an enzyme reduces that activation energy. Maybe it does it because, I'll get you a question in a second. Maybe it does it because the enzyme's like, all right, this thing doesn't want to be with this thing when you do it like this. But what if we change their shape or their direction so they're coming together like this, right? And sometimes an enzyme will do that. They make sure that those two atoms are meeting together in just the right way. So that's just one of the many ways it can do it. But yes, ma'am.
That an enzyme reduces the activation energy? Is that how it speeds up the rate of the chemical reaction?
Yes. Enzymes speed up the rate of a chemical reaction by reducing the amount of activation energy needed to start that reaction. And in some cases, not that I'm going to test you on this, but it's very important, oddly enough. In most cases, I think, in your body, most of the reactions that are happening would not happen without enzymes. Like it would require so much activation energy that your body's not going to get that much activation energy. So without enzymes, nearly all these reactions in your body would happen. So all this being said, before I take questions, here's the picture from your textbook. This is, again, this one here is from the old textbook. Here's from the new textbook. I don't like it. I feel like this is not good for a 100 level non-biology major class, but here it is. It's kind of showing the same thing, it's just being a lot more technical, right? So here's the activation of energy required to have the reaction without an enzyme, right? It's a lot of activation energy. You put in all this energy, and then finally the reaction happens and the energy is reduced. But then with the enzyme represented in the dotted lines, it requires less activation energy, right? So you get it up and then finally that's what happens. But either way, no, I don't want to see either way with that. And you can see they have the delta G, which is the change in energy. We're not worried about that, right? I'm only including that because your textbook included it. So the point here is that once again, enzymes work by reducing the amount of activation energy needed to release the reaction or to start that reaction. I should also point out, since we're talking about delta G anyway, the last reaction I showed you from the old textbook, that was an exergonic reaction, right? Because you start from high energy and you go to low energy because you released all the energy that was stored here. These things don't have as much energy as those. You released energy. This one from the new textbook is an endergonic reaction because your reactants didn't have that much energy, but something about whatever you did to them, they now have more energy. So they took energy in, right? They absorbed some energy and started off as low energy and now they're high energy. But again, more importantly, activation energy is reduced by enzymes. So let's talk more about enzymes. There'll be some questions about enzymes on the exams. First of all, enzymes are very selective. Now, this isn't exactly true, but as far as we're concerned in this class, you might as well think about it this way. As far as we're concerned, every chemical reaction in your body has a specific enzyme for it. And as far as we're concerned, you could also look at that the other way. Every enzyme in your body is there for one specific reaction. Now, that's not 100% true. You can look that up for independent work if you want. Like, what are some cases where one enzyme does different chemical reactions? Or what are some cases where one chemical reaction can be done by different enzymes? You can look that up if you want for independent work. But as far as we're concerned, they're very selective. You have one enzyme per chemical reaction. So here's some terms you need to know for the exam. Enzymes recognize something called a substrate. And the substrate is the reactant molecule. So make sure you know what a substrate is. So again, we're talking about chemical reactions, right? Enzymes make these chemical reactions happen. Well, the substrate is the molecule, or sometimes molecules, that is going to be changed in the chemical reaction. Talk about this here in a second. But for now, the next word for attendance would be sunset. So anyway, again, enzymes are very selective. What do they work on? Like I said, they do specific chemical reactions. So what is it? Which molecules are they working on? Well, it's called the substrate. How does this work? Well, the substrate enters what's called the active site. So that's where the magic happens on an enzyme, right? An enzyme, it's a big molecule, but the magic, if you want to call it that, the real work is done at the active site. The active site is where the substrate fits in, like you can see in this picture. There's the substrate, there's the active site. Very selective. So it's always that the shape of the substrate actually fits the active site. Of course, this is a generic enzyme and a generic substrate. They all have different shapes, different sizes, but this is just a great way to visualize it. So again, substrate is the reactant molecule. The reactant molecule fits into the enzyme in a place called the active site. Of course, we can see here that the shape fits, and that is also very important. But I also want to point very quickly out, it's not just the shape that fits, but it's also the chemistry that fits. So for example, if this substrate was very positive right there, then what charge do you think this part of the enzyme would be? Think about it. If that substrate's going to fit in right there, and that's positive, what charge must this be right here?
Negative.
Good, negative, right? Because positive and negative come together. So that's a chemistry fit. Or if this part was polar, then we would expect this part to be polar too, right? Because polar and polar mix together, like hydrophobic and hydrophobic go together. Hydrophilic and hydrophilic go together. Oil and water don't mix, right? So Not only does it have a shape that fits, but it has a chemistry that fits too. Of course, for the exam, that's a lot of extra information. For the exam, all you need to know is the substrate fits into the active site of an enzyme. And then what happens? Then we have something called an induced fit. So after the the substrate attaches itself to the enzyme, then the enzyme changes its shape. And then it has a better fit between the substrate and the active site. And this is why I use this picture as an example. What are those people doing?
Yes, sir.
Yeah, maybe that too. But I was going to say, at the very least, hugging, right? So you can almost think as a hug as a reaction happening in an enzyme. Because what happens is, you start off open-armed, right? Because you want to be able to get your substrate. So you start off with open arms. And then once they get into you, then that's when you bring it in for a snugger fit, right? Same with an enzyme. They're kind of like open-armed at first. Of course, this picture doesn't really illustrate that. Then the substrate comes in, and then it's a snugger fit. It's tighter. Anyway, any questions about these terms? Very important. Substrate, active site, induced fit. Actually, more so, the first two are more important. Substrate and active site. Because there's going to be some other stuff I teach you later that involve the substrate, that involve the active site. But as far as I can remember off the top of my head, that's the last time we talked about induced fit. So yes, induced fit happens. Know that it happens. You might get one question about it, but that's about it, right? There's other stuff we're going to talk about later. So any questions so far? All right, so you can see here, before I go to the next slide, the substrate came to the enzyme in the active site. You had an induced fit. It changes its shape a little bit. And then what you can't see here, but you can tell by the colors, like here, they must have broken that chemical bond to bonded this thing to that thing, right? Because now there's different colors, meaning it's no longer this one molecule. It is now these two different molecules. So that chemical reaction happens, and then it broke it apart, and now you can see the products leave the enzyme. So that's the next step we're going to talk about right here. After the product is released, the enzyme is then able to accept another substrate molecule. Again, going back to that picture we just looked at. It started empty, it did its job, and now it's empty again. So it's ready to go again. Enzymes, basically what I'm saying, are they're reusable. Enzymes are not used up in a reaction. So, I'll go ahead and tell you this right now. There'll be at least one question on the exam where I say, this thing plus this thing, and this chemical reaction gives you this thing, right? And I'm going to give you some chemical equation, some chemical equation you've never seen. Then I'm going to say, in that chemical equation, which one is the enzyme? And there's your first hint. Enzymes are not used up in the reaction. So anytime you see something on the left-hand side of the equation and the right-hand side, you know it's an enzyme. If it was on the left-hand side of the equation and then it was used up, then it wouldn't be on the right-hand side of the equation, right? The only thing that's on both sides of the equation is the enzyme. So when you get a chemical equation on your exam that you've never seen before, and it says which one is the enzyme, that's how you know, because it's on both sides. Enzymes are not used up in chemical reactions. Now take a step away back from that for a minute. We'll come back to that concept. But really quickly, I want to point this out again. So remember when we talked about biological molecules, we talked about proteins. We talked about how proteins are sensitive to their environment, because if you change the temperature or the pH, then you can change the shape. And proteins are all about the shape. But here we go again. This is what I'm talking about. So again, remember, enzymes work by that substrate that has that very specific shape in chemistry, fitting into that enzyme that has that very specific shape. So obviously, if you change that very specific shape, it cannot do its job. So again, all proteins are very sensitive to their environment. This is especially true of enzymes, because they're even more so, it's all about that shape, right? Generally speaking, so I'm going to step back to that too. Generally speaking, when you talk about chemical reactions, the warmer the environment is, the faster they happen. Because as you know from a previous conversation, when I say warmer, what do I mean at the molecular level? I mean molecules are moving faster, right? So obviously the faster molecules are bouncing around, the more likely it is for the two that need to be together will be together. right? So that's generally what happens. However, this is important, not necessarily for the exam, but for next week's lab, or I guess the next 2 weeks lab, there is an optimum range. So yes, generally speaking, chemical reactions go faster as you increase the temperature, generally. However, obviously it could get too hot. And one of the reasons it can be too hot is because of the enzyme itself. Because if you get it too hot, then the enzyme will change its shape, and then it can't do its job, right? And obviously there's also, that's what we call denatured, right? If it gets too hot, the enzyme basically melts, and then that's what we call denatured. And again, if it doesn't have the right shape, it can't do its job. Same thing with pH, right? We already gave you some examples of that in the last chapter. Put it in a bunch of positive things, you're gonna break some bonds. If you put it in a bunch of negative things, you're gonna break some bonds. So that, again, pH can change it. Salt concentration can also change it. I'm not gonna ask you any of these things. The point is, this bullet point right here. They are sensitive to their environment. There is an optimal range. And all those other bullet points are just kind of like side information, right? So I'm telling you that enzymes are sensitive to the environment, and there's an optimum range at which they work. And this other stuff is just kind of explaining why they're sensitive to their environment, and there's an optimum range. I will also point out that, not that you need to know this for the exam, but again, enzymes are very specific, right? So same with this optimal range thing. It all depends on the enzyme. Some enzymes work at really high temperatures. Some enzymes work at really low temperatures. Of course, me saying high and low is kind of a silly thing anyway, because what a human considers a high temperature might be very low to a different organism, or maybe even higher to a different organism, right? It's all relative. All right, I'll talk about coenzymes later when it's a better time to introduce them. But here's something that definitely will help you on the exam. Many enzymes are named for their substrates, and they end with the word ACE. As far as we're concerned in this course, there will be a couple of exceptions, and I'll tell you when we get there. But as far as we're concerned, you might as well just say to yourself, all enzymes are named for their substrate or what they do, and they end in the letters ACE. So again, I'm going to give you some chemical reaction that you've never seen before. I might make up some words. And I'm going to say which one is the enzyme in this chemical reaction. And remember, the first way I told you can tell is if it's on both sides of the chemical reactions, it's an enzyme because it wasn't used up. Now there's even yet another way you can tell if it's an enzyme because it ends in ACE. So if it's on both sides of the equation and it ends in ACE, it's like, oh, that's the enzyme. Does that make sense? Ends with ACE, it's on both sides of the equation, therefore it must be an enzyme. Here's where it gets a little bit trickier. For the test, again, I'll give you some made-up equation. They need to recognize the enzyme, but then I'll say, now, which part is the substrate? And this is where this last bullet point comes in. If you've already identified the enzyme, then it should be easy to identify the substrate because they're named after the substrate. So again, I'm just going to make a word up. If there's an enzyme called frothase, froth, like something you might see on the top of a certain type of coffee, right? So frothase plus something, something gives you something, something, and frothase, right? So you saw frothase on both sides of the equation. You know frothase is the enzyme. And then it says, okay, now what is the substrate? And because the thing's called frothase, you just know, oh yeah, froth is the substrate. Because again, enzymes are named after their substrate. And again, I will just make some words up. Because the idea here is for you to be able to see any chemical reaction in life and be like, yep, it's the first time I've seen this chemical reaction, but I know that's the enzyme, and I know that's the substrate. So any questions about this slide before I show you a picture of an enzyme in action? All right, so here we go. There's an enzyme. This one happens to be a real enzyme called lactase. All right, if that's the enzyme, what do you think the substrate might be? Okay. Good guess, and you were mostly correct. Based on what I told you, you were exactly correct. But of course, if you're looking at a chemical reaction, it would be anything that looks like lact, right? So in this case, it's lactose. But you'd be able to tell because that's the only thing in the chemical reaction that would be anything like lactase. So, good. Anyway, there's the enzyme. The active site's ready to go. It fits the substrate, which is lactose, perfectly. The substrate binds. It's got a nice fit. Of course, because we're breaking a chemical reaction, this is the hydrolysis reaction, therefore it requires an input of water, as we know from way back. Anyway, you got the input of water, now it's ready to go. The enzyme does its job. It breaks that lactose into galactose and glucose, and now the products are released. The enzyme's ready to go. And of course, this is just an example. I'm not going to ask you any questions about lactase or lactose or glucose or galactose. Just an example. And your book might talk about this specific reaction. When you talk about enzymes, this is one of those very popular ones to talk about, this particular chemical reaction. Just so you know, I'm not going to ask any questions specifically about lactase or lactose. But again, your book might go over it. So if you read anything in your book where it's going into a lot of details about that reaction, don't worry about it. I'm telling you right now, I'm not going to ask you any questions that specific. So that's how enzymes work. Let's really quickly talk about some ways you can modify them. That is something called an enzyme. Well, let me before I say this. If someone said, all right, let's say I was talking to you guys, they got back from Labor Day and I say, hey, look, how was your Labor Day? And you guys told me, and I said, I was hanging out with my buddies. One of my buddies drank a little bit too much and lost his inhibitions. What do you think that means? What am I saying when I said what do you think I'm saying when I said my buddy lost his inhibitions? Good, maybe. That could be one of them. How about this? Let me put it a different way. I have a 23-year-old daughter, and what if I told her, like, listen, I'm going to use this example again. Like, I don't want you drinking too much because when you do, you lose your inhibitions, and you're going to be with a bunch of strangers, and I don't trust strangers. So make sure you don't lose your inhibitions because you're kind of on the right track. The ability to do something, yes, because definitely alcohol is notorious for having you not be able to function properly. But what does something else happen? What does it mean to lower your inhibitions? Or if you even forget about alcohol, let's say, I don't know, somebody just got divorced and they go on a girls' trip and like, all right, I'm free. I'm celebrating my new singlehood. I'm throwing my inhibitions out the window. What does she mean? What is she doing? Doing more than usual. I like that. Anybody else? What did you say back there? Because I bet it was funny.
Lowering your standards.
Lowering your standards. Actually, that's closer to what I was getting at than anything else. So your inhibitions, in that sense, I'm amazed more if you haven't heard about it. I hope that means hopefully your generation is just really good at not losing your inhibitions, so you haven't had that conversation a lot. Inhibitions in that context, like what's keeping you from doing the right thing? You're lowering your inhibitions, and like, you know what?
Morality, right?
Yeah, sort of, yeah. Sometimes morality, like, you know what? I'm gonna make out with all these people in this bar. Or it could be something like, you know what? Like my dumb friends, and I'm dumber than they are, so I'm allowed to call them dumb. Like if my dumb friends, like, you know what? I know we're really drunk, but let's shoot this gun. That looks like fun, right? Not a smart thing, right? You're losing the thing that keeps you from doing something. All that being said, here's a term you need to know: enzyme inhibitors. They keep them from doing what they're supposed to do. Enzymes are supposed to do something. Enzyme inhibitors keep them from doing it because that's what inhibition is. Specifically, the way your book puts it, enzyme inhibitors inhibit metabolic reactions. Obviously, they inhibit something. What do they inhibit? They inhibit metabolic reactions. What are metabolic reactions? Those are the reactions that enzymes do. So enzymes, inhibitors, prevent, inhibit, reduce, stop, all these different adjectives you can use, depends on how good the inhibitor is. But basically, it keeps it from doing what it's supposed to do, either completely or at least not as efficiently. That's the first thing you need to know. The next thing you need to know is how it happens. And the way it always happens is because the enzyme inhibitor binds itself to the enzyme. So the enzyme inhibitor is another molecule, and it binds itself to the enzyme, which itself is a molecule, right? So these two molecules come together, and when that inhibitor binds itself to the enzyme, that's what disrupts the function of the enzyme. And I would get more specific. I'm going to do that later because there's different ways that it binds to the enzymes. And then from there, depending on the way it binds to the enzyme, it inhibits it, it disrupts it in a different way. So you're good so far with this first main bullet point right here. You know what an enzyme inhibitor is, and you know how it inhibits, right? It keeps the enzyme from doing its job. And it does it by binding to the enzyme. And that the act of binding to the enzymes is what prevents it from doing it. That being said, now I'll tell you there's two different types of inhibitors, two different types of inhibition. The first one, and you need to know the difference between the two. The first one is called competitive inhibition. And this works by the inhibitor, it is still binding to the enzyme, but now I'm going to be more specific. Where does it bind to the enzyme? At the active site. It plugs up the active site, which is what that little picture's for, right? So here you can see an enzyme. This little green thing is an inhibitor. That means it hasn't even clogged the whole site, right? Just that part. But now that substrate can't fit because the inhibitor is in the way, right? And that's exactly how competitive inhibition works. It goes straight for the source. It blocks the substrate from ever even attaching to the enzyme because it blocks the active site. So in this case, to help you remember the word anyway, it's the only reason I'm describing it this way, to help you remember the word. The enzyme and the inhibitor and the substrate are competing against each other to see which one can get to the active site first. Does that make sense? They're in competition. Does that make sense? They're both competing for the same active site. You're like having one parking spot left in Hamlin, and you're running late, and someone else is running late, and now you're coming in at the same time. Like, all right, who's going to pull in first? You're competing for the same car spot, parking spot, car spot. Whoever calls it a car spot. Anyway, any questions about this side before I tell you about the other kind of inhibition? All right, here's one you might know if you don't. Oh yes, good question.
With the competitive inhibition one, could there be like two competitive ones blocking? And then that regular enzyme one comes up there, and just two of them go in instead of just one?
You mean like using this picture as an example, as if
Like
you
two
have like
separate things.
one there and one there?
Yeah. Is that possible?
Yes. Yes, it is. And I'm trying to rack my head off of trying to remember some specific examples, but I can't. And I don't remember how rare or normal it is, but so basically, in the sense like if I were teaching it and I knew it, I would say something like, also, by the way, some enzymes have more than one inhibitors, right? Yeah. How possible it is, I don't know how normal that is, but yeah, it's normal. Be a good topic for independent work. How many enzymes have more than one inhibitor? Good question. Also, for that matter, and I don't even know the answer to this one. What if like that is a specific inhibitor because it has the right shape? What if there's some other molecule that also has the right shape that can also act as an inhibitor, even in the same spot? So again, kind of asking the same question that you're asking is, can you have more than one inhibitor for one enzyme? All right, but here's my question for you. And if you don't overthink it, I think you can get this. So don't overthink this. Right now, I just got finished teaching you about competitive inhibition. What do you think the name of the next one I'm going to teach you is? If this is competitive inhibition, what do you think the next one might be?
Non-competitive.
Non-competitive, thank you. If you send the words to me today for extra credit, remind me that you answered it, and you answered some stuff earlier too. Non-competitive inhibition. So this is when the inhibitor binds at a site other than the active site. So it is not blocking the active site. It's binding anywhere else. Non-competitive inhibition is where it binds anywhere else. And you might think, well, how does that work? If you're not blocking the active site, how does that work? Well, when it binds to something else, it changes the enzyme shape. Therefore, the active site has now changed shape. So even though it's not blocking the active site, it's changed the shape of the active site. And as we already know, because I've said it so many times, it's all about the shape, right? If you change the shape, you change the function. So when you change the shape of the active site, or the enzyme in general, right, the active site can no longer accept the substrate. Another word for this, and I'm probably not going to ask you, if I were to put it in the exam, I think I would try to put these two words together. Allosteric inhibition, aka non-competitive inhibition. They're the same thing, different words. Let's see. The last word for attendance will be, say, I don't know, I'm trying to find something. Square. It's not quite a square, but it's close. So here's some pictures of what I'm talking about, right? Here's an enzyme just doing its job, working normally. There's the enzyme, there's the active site, there's a substrate that fits in the active site, it comes in, perfect. Here is inhibition by a substrate imposter, or as we know it as competitive inhibition, right? So here we have this inhibitor. You can see the inhibitor is different than the substrate, but it's just enough to plug that site, so now the substrate can't go, therefore can't do its job. And here we have non-competitive inhibition or allosteric inhibition. Here again, the active site does not get plugged, but there's something about that inhibitor binding to the enzyme that changed the shape. In this particular case, it just made the hole smaller, therefore the substrate couldn't fit in. Either way, it's all about keeping the substrate from going into the active site, whether you're blocking the active site or changing the shape of the active site. That's a perfect place to come back, because when we come back, instead of talking about inhibitors, we're kind of sort of talking about the opposite, which are some things that help enzymes do their job. That's it. I will see you guys at 10 o'clock. Unless you need me for office hours, then I'll be there until 10. Everybody, don't forget to put some pants on or bring some pants. You'll just have to do, no, I'm going to say experiment 1, because it's not dangerous. It's just dye and hot base. Actually, none of them are dangerous. I mean, you can definitely come, and I'll just think about what to do with it, rather than like trying to figure out which one's the least dangerous or whatever it may be.
Okay, thank you.